So you'll notice the handout that I gave you at the top. Notice the title. This is a quiz over cellular respiration. The word quiz should not scare you. This is a study aid, is what it really is. You know how for every chapter we have practice quizzes in Blackboard, right? This is your Blackboard practice quiz, printed in this form. So I don't know if you've gone in there and checked out the quiz yet for this chapter. Probably not, because it's the last chapter in Module 3, and we're not there yet. But it's a 30-question quiz instead of 3 10-question quizzes like it would normally be. The reason that is it's all one topic. It's all one topic that we do. So I decided to make it one quiz, and also decided to give you something that you could follow along in class while we talk about this topic. Answer the questions, circle the correct answers. Then you can do Blackboard however you want. Go in and take the test using this and make sure you get them all right, or go in and take it after you've studied from this and see what you remember. It's up to you how you choose to do that. But everybody should make 100 on that 30-question quiz by the time you're done with it. This chapter, we do not have a lab. Today, I'll do the whole chapter with you. One big topic, cellular respiration. And cellular respiration, just to get your mind thinking about this is how you make energy in the cells. This is how you make ATP, or energy. So why would they call it cellular respiration instead of cellular energy process? Tell me what respiration means. Breathing. There we go. Respiration is about breathing. What do you take in that your body really needs from the air? Oxygen. So check this out. We're going to talk about how your body uses oxygen and glucose, or nutritional elements like glucose, to make ATP, to make energy in your body. All right. Now you may remember you've talked about ATP in AMP1 here and there, little bits. Do you remember doing muscle contraction at all, or studying the inside of the muscle? I don't know if your teacher did this, but when I taught AMP1, I would always do this. There's a molecule called myosin, and actin and myosin interact, and that shortens the muscle. Look, pretend I'm the myosin right here. I have little heads right there, and those heads attach to the actin and pivot. Now, when I do this in AMP1, I have 2 students come up, and they would be actin, and I would slide them past my little myosin heads here. Here's what I want to give you, though. In a muscle, you have lots of myosin, and those little heads are all over the myosin. There's hundreds of them on each myosin, and there's hundreds of myosins in a myofibril, and there's hundreds of those in a muscle cell. So we're talking hundreds of thousands or millions of these little heads in your muscle, in just one little tiny muscle. One head. It takes 2 ATPs to make one head attach, pivot, and release. It takes 2 ATPs to make one head attach, pivot, and release. Okay? So what if you had eaten a cracker and you got 10 ATPs from it? What could you do with that ATP? Attach, pivot, and release how many times? 5 times, right? So when we do this, and what I'm telling you right now, I'm not going to ask you this on the test. This is just to give your mind something to consider. When you hear ATP, I think muscle contraction is the easy thing for us to visualize. Think about those little heads, and the ATP is the energy that makes those heads attach, pivot, and then let go. Okay? It takes energy to do that. So now we're ready to talk about the processes that make the energy, how it happens, and I'll come back to this when it's relevant. Cellular what? All about creating what molecule in the cell? ATP. Today I'm not going to show you the structure of ATP and go through it as a molecule, but it is a cool little molecule. very close to what you find in your DNA. Little pieces of DNA are very close to this. This process has many steps. It actually has a few different processes to the big picture. So what we're going to do, as you can see from your handout, is we're going to categorize this into the various processes of cellular respiration and do them one at a time. So who wants to tell me, from looking right up here, what's the first process in cellular respiration that we're going to talk about? Yeah, it's called? It is called glycolysis. It is called glycolysis. Anybody in here have Dr. Day?
I have.
Yeah. Remember how you love to break down words? Well, the same. Glycolysis means something. It means something. Anybody remember from AMP1 what lysis is? Breaking down. Good. Cell lysis, to tear apart a cell, to lyse something that's a molecule, you're breaking it down. What do you think that might be? Sugar, or in this case, glucose. Anytime they use glyc, they're referring to carbohydrates or sugars, and sometimes it's glucose, sometimes it's others, but it always means a sugar. So the whole idea, when we're going to make energy, we start by breaking down sugar, a sugar molecule, okay? And so glycolysis means the breakdown of sugar. In our case, we're going to start with glucose. So I'm going to write glucose right here. The pen is a little funky still. Okay, next step. Anybody remember your chemistry lab from AMP1? Notice I don't see any hands because they don't want to be called on. It was a long time ago. And when you have an 8-week class, you do chemistry like in one day. It's hard for it to stick, okay? No matter who teaches it to you. So I'm going to remind you something about a glucose molecule. Here's its formula. That tells me we have how many carbons? 6. How many hydrogens? 12. And how many oxygens? 6. Okay. Hey, what is... That's like H2O, right? How many of them would that be? 6. That's 6 waters right there. And this is 6 what? Carbons. I don't know if you remember this from AMP1, but the reason we call those molecules like glucose a carbohydrate is because hydrate means water and carbo means carbon. They're basically carbon and water mixed together in a certain way to create a molecule. Okay. With this, I don't expect you. Now let me rephrase that. We're not going to follow everything in the molecule, but what we're going to do is we're going to watch the carbons. We're going to keep track of what goes on in cellular respiration by keeping track of the carbons and watching the carbons. It'll become clear later why we do that, but for right now, notice how we're going to do this. When we mention a molecule, a starting molecule, or what it becomes, I'm always going to let you know how many carbons in that molecule. So to begin with, glucose, how many carbons? 6, right? And we're not going to worry about the rest of it. We're just going to say, okay, glucose is a 6-carbon molecule. Well, in glycolysis, what the cell is going to do is break down that 6-carbon molecule and turn it into 2 3-carbon molecules. It's going to convert it into 2 3-carbon molecules. Now, we don't need that in our notes, so I'm just going to erase because I need the room. And the molecules that we produce are called pyruvic acid or pyruvate. You could name that either way. So probably the next time I write it, I'll write pyruvate so you can see the spelling of both. Okay? So once again, glycolysis is the breakdown of glucose. We basically split it in half, It is not one step. It's like 10 steps to tear this molecule apart, okay? We don't need all that. This isn't biochem. This isn't cell biology. Heck, this isn't even general biology. We do it a lot different in here. We're going to understand that that molecule, through this process, is used to create two smaller molecules, okay? This happens in the cytoplasm. That just means generically in the cell. It just happens in the cell. There are chemicals in the cell that allow this to take place. When we do this, remind me why we're breaking down our sugar in the first place? What's the whole goal today? Yeah. To make energy or ATP. So every time we can, we're going to talk about, or every step of the process, we're going to talk about the energy. How much energy do they get out of this process? And so, pretty neat. We gain, very careful in my language, we gain 2 ATPs worth of energy from doing this. So by breaking down the sugar molecule, the glucose molecule, and converting it into pyruvate, going through all the steps to do that, there is enough energy released from the molecule to create 2 ATPs, 2 additional ATPs that we did not have, okay? That's cool. Another neat thing is this. There's a little extra energy that's produced. And it is another molecule, a different molecule, and we're going to call it NADH. You do not need to know the long for what it stands for, okay? We're just going to go with that. What's an H, though? What do you think? Hydrogen. Okay, that's a hydrogen. And the NAD part comes from one of your B vitamins. You may have heard of niacin before. B vitamins are very important to energy. So this came from a B vitamin and hydrogen combining and some other stuff. Anyway, we like to do it this way. We go, we just kind of say NAD is going to be converted into NADH. And sometimes we might even say ADP is going to be converted back into ATP. Does anybody remember what the T in ATP stands for? Tri. What does tri mean? 3. The P is phosphate. So ATP is a molecule that has 3 phosphates in it. What do you think ADP is? Adenosine diphosphate. What would di mean? Only 2. So here's what I want you to see, not trying to get technical with the chemistry, but to say, oh, the way we make the energy molecule is we take a smaller version of it that only has 2 phosphates. That's what the P is, stands for a phosphate group. And we just add a phosphate to it. Through this process, we're going to add phosphates to a molecule that's already in your cell. It's been used before. The body's a great recycler. So this side, really not going to ask you about this side on the test. It's this side that I want you to know. These are the molecules that are produced from going through the process. I wanted you to see that they had somewhere they came from. They weren't just created from scratch. They came from something that was already there. But this is the final over here. That's what we've done with the glucose. And here's the energy that we've gained from it. Oh, by the way, I'm big on funny little abbreviations. When I put that, that just means energy. Those molecules are energy molecules. This one, later on, the mitochondria is going to be able to turn that into ATP. So every time we get these other energy molecules, like NADH, we're going to be able to convert those into ATP or usable energy later on in the process. It's going to be the last step we talk about. Okay? All right, so let's check out where we are. What was our starting molecule in all this? Glucose. Glucose, which is a sugar, right? Where did we break that glucose down? Very good. In the cell, in the cytoplasm of the cell. And during the process of breaking it down, we're trying to get some energy from it. We're getting energy, converting that energy into molecules so the cell can use that energy. How many ATPs worth of energy do you get from this first step, glycolysis? 2. How many other energy molecules did you make? Oh, I see what you're saying. I asked that a funny way. You're making 2 other molecules, and what are they? They're NADH, right? It's one molecule, but you're making 2 molecules of them. So that was a weirdly worded question. All right. Remind me where this took place. Cytoplasm. So that's just inside the cell. Keep this in mind. This isn't happening in the juice of your blood, okay? This is happening in your cells. Your cells do this. Your cells create energy. Now, here's a really cool thing about this. This process can happen with or without. What's that molecule? Oxygen. Whether your cell has a good supply of oxygen or not, this process, this one step can happen. Here's what this means. If you run 100 meters, you sprint, and you've depleted the oxygen from your muscle cells in your body, you can still do this because it doesn't need oxygen. It also means if you're sitting right there right now and your cells are fully oxygenated, they can also do it because it doesn't rely on oxygen at all. Doesn't need it. You can have it or not have it, and you can do this. But remind me, how many ATPs do we get from this? So from one sugar molecule, we get how many ATPs right here? 2. How far, if you only had 2 ATPs of energy, how far could you walk? You could make 1 myosin head in 1 muscle, attach and pivot 1 time. That would not even produce perceptible movement, okay? This, breaking down 1 glucose to get 2 ATPs, that is very low on the energy scale, all right? You're gonna eat a piece of pizza that has a lot of glucoses in it, a lot of protein, a lot of fat, a lot of carbohydrates, and you're gonna get a lot of these. So we eat a lot of food because we need a lot of energy. Okay? So fancy name for a process that does not need oxygen. Very good. It is an anaerobic process. So sometimes they will say, oh, glycolysis is anaerobic respiration, okay? Because it's part of cellular respiration, but it doesn't need oxygen. So it's known as anaerobic, all right? So what's our process? Yeah, there's 2 answers because I didn't ask a specific question. So the whole thing is cellular respiration. What's the first step of cellular respiration called? Glycolysis. Why is it called glycolysis? Perfect. We're breaking down sugar. We're breaking down sugar. To do what? To produce energy. Very good. So go to your handout. I'll put it up there. See if you can answer question 1. Where does glycolysis take place? Cytoplasm. Circle it. Circle it on your paper. I would. Number 2. How many CO2s are produced as a waste product during glycolysis? Y'all, some smart people, because there's a reason I haven't said CO2 yet, because we didn't make it. If we would have made it, I would have mentioned it. So let's go back to the picture and let me show you why this answer is 0. How many carbons did we start with? 6. And we made how many molecules? 2, and each of those molecules had how many carbons? 3. So what's our total carbon count here? It's still 6. We have retained every carbon. In the next step, I'm going to start showing you what happens when we change the number of carbons. And what happens is we produce a CO2, okay? But right here, we did not produce a CO2. So the answer for number 2 is 0. It's always a weird question because I get to that question before we've talked about carbon dioxide. but in the other steps, we will be talking about carbon dioxide. So I like to ask the same questions in every step to get your mind thinking by certain processes. Number 3, how many net, it's just a fancy way to say overall gain of ATPs. How many net ATP molecules did we produce or gain from glycolysis? We gained 2. We gained 2. Number 4, how many NADH molecules did we gain or produce from glycolysis? 2. Good. How did we classify glycolysis? Anaerobic. Why? What did anaerobic mean, Annalyn? Without oxygen. It meant without oxygen. Okay. Number 6. Glycolysis can occur when oxygen is present, even though glycolysis is anaerobic. True or false? That is very true. Very good. That is very true. Number 7. Ooh, now we got to think about it. The final products from the glycolysis of glucose include. I heard 2 people say 2 things, and they were both correct. We make 2 pyruvic acids, and we produced 2 NADHs. What else could I have put there that I didn't put that would have been correct? 2 ATPs could have been in that question. It just wasn't, okay? So just know, this quiz is a quiz. It is not a statement of all necessarily true facts. It is a quiz to get you thinking. This question could be changed to include ATP to see if you know how many ATPs are made. Okay, keep that in mind. And then by the way, C and D, you have not heard of those yet. Okay? At this point, you would be, why is he throwing stuff out that we haven't heard of? Because you're going to hear of them later. Number 8, when oxygen is not sufficiently present within the cytoplasm during glycolysis, the NADH will do something. It will get rid of its hydrogen. And this hydrogen will be picked back up by pyruvic acid, and it will become something else. Okay? So here's what I'm telling you. This process does happen without oxygen, right? But for us to continue to break this down and go through the normal process, the future processes will need oxygen. And so what happens if in this step you don't have oxygen, you can't move forward. So the NADHs that get produced work together with pyruvic acid, and it produces something called lactic acid. So that's your answer for number 8. It produces lactic acid. I'm not going to put lactic acid on the board. You just have it on your quiz, and I'm okay with it only being there. Once again, if you didn't get it yet, there's picture 1. I'm going to go ahead and snag it. Okay, and the way that this process is going to work then is this. Whatever molecule we're left with, right here, that's our starting molecule for the next step. So we took glucose and we broke it down into, remember I could call this 2 pyruvates? We broke it into 2 pyruvates. And so now in the next step, we're going to start with 2 pyruvates. So I'm going to take everything off there. Who can remind me how many carbons for a pyruvic acid or pyruvate? So there is your pyruvate molecule. This next step, we are going to do this. Before I even give you the name of the molecule we made, tell me what that is telling you. Yep, we've lost 2 carbons. We've lost 2 carbons. So when we break down pyruvate, we're going to break it down in such a way that we remove some carbon from it. For each molecule, we're going to take 1 carbon away. So here we had a total of 6 carbons. Now we only have a total of 4 carbons. The molecule that we produce is called acetyl-CoA. The CoA stands for coenzyme A. I'm not going to require you to write that. This is a lab test. So just know if you see coenzyme A, don't be fooled. It's just acetyl-CoA's longer name. All right? Usually they say this happens in the mitochondria, but it could happen a little bit in the cytoplasm. Either way, I'm not really going to ask you where this step takes place. It's just in the cell. After this, it becomes more important about the mitochondria. What is our whole purpose in continuing to break down the molecule that began as a sugar? To make energy, hopefully in the form of ATP. But thankfully for us, we have another energy molecule. Who wants to tell me this name? Remind me how we pronounce that? Yeah, it's not hard. NADH. So this step right here in the second step, and it has a couple of different names. I'll tell you about the name of it in a second. All we get energy-wise is 2 NADHs. That's it. This step does not produce ATP. It simply produces 2 NADHs. Okay? Now I'm going to give you a little more on NADH this time. I like to call NADH a high energy molecule. Who remembers what we're going to do to this later on? Yes. Later on, we are going to make ATP from this. And I call it high energy because it's going to be able to make multiple ATPs. We'll talk about how many later on. But it's going to have enough energy to make multiple ATPs, not just one. So it's a pretty high energy molecule. It's carrying a lot of energy, which is really cool. Those 2 carbons that got removed, the way that we get rid of carbon out of the body is we attach it to oxygen and we turn it into a gas. And then we put it in our blood and we breathe it out. That respiratory system, right? When we breathe in, we're hoping to get oxygen. When we breathe out, we're hoping to get rid of CO2. So the cool thing is, whenever we change the carbon count, we make CO2 or carbon dioxide. That CO2, those 2 CO2s, are considered a waste product from this, okay? So in cellular respiration, we are going to be trying to make ATP, and during some of these steps, we are going to produce some carbon dioxide. How do we get rid of the carbon dioxide? We breathe it out. We breathe it out, okay? Since this process, that we attached an oxygen to the carbon, it used oxygen. See, if we didn't have oxygen, we couldn't make CO2. We would make C something else. We couldn't do that. So this process is aerobic. What does that mean? Needs oxygen. It absolutely needs oxygen. It seems like every book that talks about this process gives it a different name. It's kind of an intermediate step. So some books call it a transitional reaction or something. The process, as you can see on your handout, I refer to this as oxidative decarboxylation. Let's go with the Dr. Day way of breaking down words to help us understand what that means. Now, this isn't pure biochem. I'm giving you a memory device here. I'm not giving you true technical biochemistry because oxidative doesn't necessarily mean this. I'm giving you a way to think of it. What word does oxidative make you think of? Oxygen. What are we using in this process? Oxygen, along with our byproduct pyruvate, right? So that means we are using oxygen. And we are using oxygen to get rid of what right there? Carbon. Do you know what D means? Removal. D always means removal. To get something out, take it away, or remove it. So the name oxidative decarboxylation can tell you you're using oxygen to get rid of carbon, okay? That's a memory device. That's not the technical, because technically, decarbox means we're removing carbon and oxygen at the same time, okay? So, and it's an oxidative process, which is about electrons. There's step 2. Go ahead and snag your little step 2 picture. Who can tell me how many ATPs were produced? 0. 0 ATPs in that step. So let's go on our handout. Notice this transitional phase, I only gave you 4 questions. It's not as intense a phase. We don't need as many questions to understand it. Number 9, how many CO2s were produced as a waste during this step? 2, very good. Number 10, how many ATPs were made during this step? 0. Okay, we did have some energy. How many of those NADHs did we produce? 2, very good. And at the end, over here, what are our products? Acetyl-CoA and NADH. How many acetyl-CoAs? 2. How many NADHs? 2. Very good. Very good. So far, what have we used oxygen to do when we're making ATP? To get rid of carbon. Exactly. That's why you breathe. You breathe because without getting rid of the carbon, you cannot continue this process. You need that oxygen to remove that carbon from this molecule. That is a very important part of the process. So the reason we're breathing is so we can keep that chemical reaction going in order to produce our ATP. Okay, so why did I do it this way this time? Well, this is what is left of our sugar right here. We had 2 molecules that were each 2 carbons. What do we call it? Acetyl-CoA. Here's what is going to happen next. We're going to go into the mitochondria. Okay? We're going to go into the mitochondria with this molecule. And once it enters the mitochondria, keep this in mind, your mitochondria have been in those cells for a long time. They're not brand new. They've been there. And they've been doing this for you for a while. So, whenever you made energy before, this step produced some byproducts. Okay? Those byproducts, those chemicals, are still in the cell. One of those chemicals has a crazy name. Oxalo, I guess I'll have to rewrite acid. Oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid happens to be, tell me what that means, a 4-carbon molecule. Okay? And it just so happens that we're going to have 2 of them, at least, but we're going to use 2 of them. 2 of them that have been sitting around there we're going to use. Okay? Why are we going to use 2 of them? How many acetyl-CoAs do we have? We have 2. It's 4 carbons, but we have 2 molecules, right? We have 2 of those, so we need to pick up 2 of these. We need to use 2 of these. Here's how this next step in the mitochondria, here's how it works. Those acetyl-CoAs that you see right there, they're going to be enzymes that allow them to join this molecule that was left over from last time. So our 2-carbon molecule is going to join a 4-carbon molecule. Y'all do the crazy math. What's 2 plus 4? Wait, let me ask the whole class. I only heard one person doing math. What's 2 plus 4? 6. So we are going to make, in this step, we're going to create 6-carbon molecule. That is called citric acid. Anybody want to take a wild guess how many of those we're going to make? 2. Because we had 2 acetyl-CoAs. Each acetyl-CoA is going to join an oxalo, and from each of those we're going to produce a citric acid. This is, remember, this is just what happens. As we break down the sugar and get it smaller, and we got it to a 2-carbon piece, when it goes into the mitochondria, and by the way, this thing's called the Krebs cycle, and when it goes into the Krebs cycle, what those 2-carbon molecules do is they are going to join a 4-carbon molecule that's already there. And it's going to create a brand new 6-carbon molecule that they chose to name citric acid. And here's the fun part. We are going to then kind of play around with that molecule, break it down a little bit, reconvert it a little bit, do some things with it. Now, I'll take that last arrow off. We don't need to try to be correct with how many steps it is. We're going to take that citric acid and break it down until we make it what? Go ahead, let me say it again. We're going to take citric acid and break it down in the Krebs cycle until we turn it into what? That oxaloacetic acid. That's where that oxalo came from. What the Krebs cycle does is it works on that citric acid molecule to tear it apart and get energy from it. And when it's done with it, it turns it into what we're going to need later. It just leaves it there. Okay. This process, step 3, is called the Krebs cycle. Sometimes they'll call it the citric acid cycle because that's our beginning molecule here. What is our goal in tearing down the molecules we produce? What are we trying to make? Energy in the form of hopefully ATP, and if not, we'll take that energy and put it in another high energy molecule to hang out and wait until we can make some ATP from it. So you ready for this? Are you kidding me? All the energy we get from this in the form of ATP is 2 of them. That's it. We only get 2 more ATPs. Who can remind me, glycolysis, how many ATPs in glycolysis? 2. Did that second step, that transition step, did it have any ATPs made? Nope. And now we get to the third step. By the way, there's only one more step after this. We get to the third step, and how many ATPs do we make? So up to this point, three big processes coming through here to make energy, and we only make enough energy to make that myosin head attach, pivot, and release twice. We only make 4 ATPs, okay? 2 here. Good news for you. This step makes a lot more of the high energy molecules. It actually makes 6 NADHs. Makes 6 NADHs. And by the way, after this, I'm going to go show you some fancy pictures from the publisher that gives you a little more insight into why this is made. Hey, if you see that part right there, what are you thinking this FADH2 molecule might be able to do? What do you think we might be able to do with those later? Generate ATP. Good answer. Generate ATP. So here's how you say its name, the abbreviated version, FADH2. We make 2 FADH2s in the Krebs cycle. These are also just like in our previous step, NADH and FADH2. Later, Later they'll be what? They'll be converted into ATPs later on. Now I can show you how to count this number so you don't mess it up. How many carbons total here? 2 times 6, 12. Over here, 2 times 4. What is 12 minus 8? We're subtracting 8 from 12. We get 4. Whenever we have extra carbons that we removed, what do we turn them into? CO2, our waste product. So from the Krebs cycle, which takes place where? In the mitochondria, we get directly how many ATPs? 2 ATPs. But we make 2 other high energy molecules, right? We make 6 of those FADH2s. We never made that many before. Or sorry, NADHs. And we make 2 of our molecule called FADH2. FADH2 it comes from a different B vitamin. You may have heard of riboflavin before. It is an energy molecule made from that, and it doesn't have as much energy as NADH. It's less energy, but I'll show you that in the next step. But still, it's a high energy molecule, and later on we'll make it into ATP. And remember, our whole purpose for using oxygen is to remove what? To remove carbon. So because we have 4 carbons that got torn off, we have to get rid of those. We make 4 CO2s. So that is your Krebs cycle. That is your Krebs cycle. Would you call it aerobic or anaerobic? Why? It needs oxygen, yes, sir.
The equation looks like it's balanced. Should be like the finished product is citric acid.
So the final product, oxaloacetic. Okay. So when we finish this, what we're left with is oxaloacetic acid and these things. Okay. Because we have to. This is our starting molecule. Okay. This is our finished molecule right here. So let's look at this before we go look at another picture. Part 3, the Krebs cycle. Oh. Let's see if you can guess. How many times does the Krebs cycle have to occur? We say spin, because notice it's a circle. Okay. How many times does the Krebs cycle have to occur, spin, for each glucose that is fully broken down? Has to happen twice. That's why every time we've been doing this, remember, we started with 1 glucose, but that 1 glucose became 2 pyruvates. And so now the main molecule, there's always 2 of them. So when we get to Krebs, the Krebs cycle happens twice. That's why I keep putting those 2s. From the 2 spins of the Krebs, this is what it's made. Every question I ask you will be about Krebs fully completing, and Krebs needs to happen twice. So I will always phrase it that way. I won't cut it in half or try to make you think of it in another way. So the answer to 13 is 2. How many CO2s did we make as a waste product here? We made 4. How many ATPs did we make here? 2. There you go. How many NADHs did we make here? 6. Wow. Turn the page. Krebs cycle is considered aerobic. Why? Okay, we used oxygen to get rid of carbon. Very good. And number 18. Wow. The products that result, or I could say that remain, at the end of the Krebs cycle include what? There you go. And don't forget this one. The 2 oxaloacetic acids. So the answer is, notice I did not try to confuse you by changing the numbers. That's not what this is about. It's not trying to be tricky with the numbers. This is about helping you see that when we're done with Krebs, we made 4 CO2s we made 6 NADHs, we made 2 FADH2s, we were left with 2 oxaloacetic acids that we can use later, and we did make 2 ATPs worth of direct energy. Okay? So there is your picture. There is your picture. Well, let me put one word over here. That's where we consider Krebs starting, when we tear down the citric acid. So this is as far as we take that glucose molecule. That sugar that we had at the beginning, remember, this is kind of interesting. At first we tore the sugar down and made it 2 3-carbon molecules. Then we made it 2 2-carbon molecules. And then we put it back together. And in this step, we're left with a 4-carbon molecule. So we just kind of have kept messing with that molecule. Okay, how many ATPs did we make in glycolysis? 2. How many ATPs did we make in Krebs? 2. So notice, please note, every time we've made ATP, when we're breaking down the sugar molecule, we only make 2 of them. Okay, we only did it twice, but we made 2 of them each time that we did it. Okay, in every step so far, which high energy molecule did we also produce? Every time, did you notice that? First step, second step, third step. Every time we made NADH. How many did we make the first time in glycolysis? 2. How many did we make the second time in the transition? 2. And it wasn't until Krebs where we finally got away from that number. And then in Krebs, we made a ton. We made how much? 6. Okay. Any other step did we ever hear FADH2? No. This is the only step you make it. Okay? So this is something new. And because of the amount of energy that's produced and just the way that it goes, they couldn't make another NADH. There wasn't enough energy to make a whole NADH, so they make a couple of lower energy molecules. That's why this is done. Down there. And once again, every step that is aerobic, you can tell it's aerobic because what do you produce as a waste? CO2. If it's aerobic, up until now, we've always made CO2 as a waste. Okay? This is the bulk of cellular respiration. This is how we make our ATPs. This is what we do with the molecules we eat for food. So if you're eating carbohydrates and you're tearing down the sugar for energy, you do this. Hey, what if you're keto? What if you're burning fats and protein? What if you're not getting any carbs? Your body can break down proteins into little carbon pieces and plug them in. If you're breaking down fats, your body can take your fats and break them down into little carbon chains and plug them in. So the reality, I don't need you to be biochemists, I just need you to understand. Whether you eat more carbs, fats, or proteins, your body can create energy by using this process for all of those. It is just traditionally the way society goes. Most of us burn carbs for energy. Most of us eat a lot of carbs. We have excessive carbs and we burn that for energy. So that is how we teach this based off the glucose molecule. Okay? Crazy thing. You could eat no sugar at all. Eat only protein and fat and no carbs. Your body would make sugar for you. Okay? So your body would actually produce sugar, so you would still have sugar for this in addition to breaking down amino acids and fatty acids. So big picture concept, and notice none of that was on here. Why? Because what I just told you is not test questions. It's just common sense knowledge that's not so common, so I'm saying it just so you know. Carbs, fats, and proteins. Yes, you can break them all down for energy. We just teach cellular respiration based off glucose. Okay, that's the traditional model. All right. So now, what do we have left? Oh yeah, if you looked ahead, you've got a little sheet that shows you where we're going. The next step is also in the mitochondria. It's got a fancy name, but here's what it's about. We have to take these. And do what with them? What are we going to do with those NADHs and FADH2s? Now we need to make some ATP from those. We don't just leave those high energy molecules laying around. We need to use them to make some energy. So let's leave this for a second and let me take you. So here's a summary of this picture, and you are welcome to take a picture of this if you want to have it in your notes. It just might help you understand it. But look what it says. Whether you're eating proteins or carbs or fats, you're going to take those molecules and you're going to plug them somewhere in to cellular respiration. And going through these processes of glycolysis, the transition step that made acetyl-CoA, and then the Krebs cycle, we're going to get some ATPs from that, right? During those processes. So that's just an overview picture. This is the Krebs cycle. I'm gonna, because our projector's so weak, I'm gonna turn the lights off. How many times, do you guys remember how many times did the Krebs cycle have to happen for our one beginning sugar? 2. Why did it have to happen twice? Why does it happen twice, not once? What did we do with sugar in the first step? Break it down and turn it into how many molecules? 7, 12, 42, 500? We took, what was the first part of cellular respiration called? Glycolysis. What did we lyse or break down? Glucose. And what did we convert it into? There you go, pyruvic acid. Who can tell me how many pyruvates or pyruvic acids did we make? 2. So let's think about this. We took glucose and we turned it into 2 molecules, 2 separate molecules. And then we followed those through. They went from pyruvate to acetyl-CoA, and then they went into the Krebs cycle. And because we had broken down glucose into 2 molecules, now we have to make the Krebs cycle happen twice. Okay? Because glucose got turned into 2 molecules, we had to make the Krebs cycle happen twice. That's why. So here, I just want to walk you through. These details are not needed for you on the test. I just want you to see, so concretely you'll know, why did we get 6 NADHs? Why did we get 2 FADH2s? Okay? How did that happen? Where did it occur? Can you guys read? What's this molecule over here? That's our citric acid where we started. And what are we left with over here? Oh, thank you. Oxaloacetic. Yeah, oxaloacetic acid. So this whole thing, this is our Krebs cycle happening, right? We started here and we finish over here. So here's what I want you to note. As we go through the steps at a certain part in breaking down the molecule, we make an NADH right there. And then in the next step, we make another NADH. And then we have to go a while all the way to over here before really we make another one. So as you're looking here, you're seeing 3 different times we made an NADH. Since this happens twice, that gives us our 6, and let me use a black pen for that. That gives us our 6 NADHs. Okay? If we come over here and do it again, going down, going down, going down, going over. One time we got a FADH2. Since Krebs always happens twice, that gave us a total of 2 FADH2s. Okay. These little black balls, what do you think they're supposed to represent? Oh, nice. They're carbon. They're carbon. There's 6 of them here. Oh, there's only 5. And now as we come over, now there's only 4. And we finish up at 4. So we've lost 2 carbons. We've done it twice. So we've produced 4 CO2s. And now the most important thing. Why do we do all this? To try to make what? To try to make ATP. Look at that. The whole Krebs cycle, 1 ATP. But we spun Krebs how many times? 2. 2 times. 2 times. It happened 2 times. So because of that, we get our 2 ATPs. Oops. 2 ATPs worth of energy. So there are our energy molecules and our waste product. How did we get rid of CO2? Well, how do we get it out of the body? Oh, we breathe it out. So now we know what happens to that. Okay. What are we going to do with these ATPs? Use it for energy. Yeah. So the only thing left that we need to look at is we got to process these right here. Now we need to process those. Plus the NADHs from the other steps. Okay? That's what the last step, the electron transport chain, is all about, processing these high energy molecules. If you want the picture, because it might help you to understand it, go ahead and snag the picture with the whole Krebs cycle up there. It's up to you, okay? Where did the Krebs cycle take place? Very good. In the mitochondria of your body cells. Okay, in the mitochondria of your body cells. You probably even remember in elementary school. Oh, look, there's the mitochondria. What did you call the mitochondria way back in elementary and junior high? Perfect, the powerhouse of the cell. Exactly. We call it the powerhouse of the cell. Now you know why it's called the powerhouse of the cell. What is it producing? ATP, energy, ATP, right? Here's the shape of the mitochondria. It looks like a little bean, but notice it's got its membrane out here, but look what it also has on the inside. It's got another membrane. So it's got a very special membrane that's really highly folded, that's throughout. So mitochondria has a couple of different compartments. It's got a little space out here, It's got a ton of space in here. So it's got a couple different spaces. No big deal, we'll get to that and we'll see that. Aha. I don't want to use that one yet. There's the picture I was looking for. Right there. Zoom it up. Zoom it down. Try to get it just the right size. There we go. And thinking back to AMP1, cells and chemistry, anybody tell me what these things are supposed to be right here? Yeah, that is the phospholipid bilayer. Okay. Now you may remember that cells on the outside of the cell, they have a phospholipid bilayer. So I could come up here and I could go, just to remind you, So there would be a cell membrane right there. This is the membrane on the inside of the mitochondria. Remember I showed you that picture? They got all those folds. So here's the inner membrane. So interesting thing about the mitochondria, it has two membranes, and between these two membranes, we now get this space right here. And that space is a certain size. Okay? If we push things into that space, if we pump things into that space, we create a pressure gradient or a concentration gradient. Think of it like this. I have a balloon here. If I pump air into that balloon, have I increased the pressure in the balloon? Yes, right? If you blow air into a balloon, you increase the pressure. Because the balloon's outer skin is flexible, it expands. Our mitochondrial membranes are pretty tight. So they're not really going to puff up and expand that much, but what it's going to do is it's going to increase the pressure in there. Just want to mention that even before I get started. Okay? Name the entire organelle that we're seeing on the screen right there. What is that thing? The mitochondria. Okay, the mitochondria. And yes, there's different parts of it, but you can't forget that that's where we are. We are in the mighty mitochondria. And our job now is to take all of those NADHs, and what was its little cousin called? FADH what? 2. FADH2s. And what are we going to do with these little high energy molecules? Convert them into ATP. So that is our job now. So what I'm here to help you with now is this is the last step. You can see it on your paper. It is called the electron transport system, or sometimes it's called the electron transport chain. Okay? ETS or ETC. Either way. Electron transport system, electron transport chain. I need to let you know that these molecules that contain energy, they have their energy in 2 locations in the molecule. The actual molecule has 2 locations where it has energy. Here's the answer to where the energy is in these molecules. Got to pick something bright enough to see, but I want it to stand out. The energy will be, let's put it, give me a little room to write it right here. That is where we're going to get the energy. The energy that we're going to use for the ATP is going to come from 2 places on those molecules. It's going to come from their hydrogens, and those hydrogens are not charged, so those hydrogens are carrying electrons. The second part of where the energy comes from is it's going to come from those electrons. Okay? So I want to make sure that you guys understand. Whether it's an NADH or an FADH2, those molecules carry energy in 2 places. They're hydrogens and the electrons that are on those hydrogens. So hydrogen and electron. So right now for me, let me see if I thought I had that question built in here. Oh, there it is. Number 23. Go to number 23 on your handout. The energy that is used to form ATP from NADH and FADH2 comes from which sources? Go ahead, speak it. Hydrogen, electrons, see number A? You see A? Do y'all know what a proton is from A&P1? These days, a lot of people come from A&P1 and they forget a proton is another name for a hydrogen ion. So on your handout, A is correct also. But let me tell you something. When I created the Blackboard quiz, I've been using this quiz forever. When we went from 16 weeks to 8 weeks, we dropped some of the chemistry details. So I changed the question. So your Blackboard question, A will not say proton. So when you're taking this quiz in Blackboard, question 23, B will be hydrogen, C will be electron, but A will be something different. I think I might have changed it to water, or I don't know what I changed it to. But either way, A will not be a correct answer because it won't say proton, okay? This is not me trying to be tricky, it's me changing something from before, okay? And yeah, anyway, so that's that. So when you take the Blackboard quiz, you can just scratch out proton if you want, and just know it's going to be hydrogens and electrons. All right, next thing. I guess I need to show you how the hydrogens and electrons create energy. So here's an NADH. Here's a FADH2. They move into the deep portion of the mitochondria, and here's what happens. They release their hydrogens, and that membrane will push them across. So notice up here, we have our hydrogen ions, our little hydrogen ions all in there. Because they are pushed across there, they are now pressurized, so to speak. Look what happens over here. Over here, those hydrogens that are under pressure move back through a special channel. Calling it a channel now, I'm going to change that. So it moves through a special channel. And that special channel over there uses the energy from the hydrogen shooting through because it was pressurized, so that hydrogen moves through. And the molecule, literally on a microscopic scale, it is shaped like that. It looks like a little crank. And so one spins and it spins the other. And what it physically does is it takes molecules of ADP that had only 2 phosphates. The little phosphates are hanging out here. It collects them and it puts them together. And the fancy word is it phosphorylates. It adds a phosphate to the ADP. And it creates ATP over here. So there is a little machine in your cells, a little chemical molecule that physically moves when the hydrogens come through, and it adds phosphates to a molecule to make your ATP. Since it is creating ATP, that molecule's name is ATP synthase. Synth for synthesis, create. ACE, anytime a molecule has ACE on it, do you guys remember from A&P1 what's that mean? ASE on a name means it's an enzyme. Any molecule that you see end with ASE, like ATP synthase, it's an enzyme. So this little thing over here, we could call it a pump. We could call it a channel because it's got those parts. We could also call it an enzyme, okay? And its name is ATP synthase. That is where the ATP is actually made. So I'll say it once again so you've heard it. The energy from the hydrogens is due to their pressure or concentration gradient. And when they get moved through this ATP synthase, its energy transfer is used to create ATPs. So that's where we get the ATPs. Okay. The next step. Electrons, as the hydrogen is pushed up here, notice those hydrogens have positive charges. So that means they must have given up their little negative molecule, which is the electron. And what you can see here is the electron gets pushed around through these various molecules along the membrane. Yes, this is the inner membrane. but those electrons get moved along that membrane. This is the fun part that I like to do. I know this is boring. There's a light. How do we make a light come on? Turn a switch, but what happens in the switch? What does it allow to happen for that to occur?
Electrons.
It allows electrons to flow. So what we're doing is we're blocking them from flowing, and now we're allowing them to flow. So what you may not know is electricity is primarily electrons moving. So on your membrane here, when we move the electrons, that's energy. And these little molecules capture that energy and use that energy. So two forms of energy come from the hydrogen ions that creates a concentration or pressure gradient, and the electrons that move along the membrane. Okay? That's where we get energy to create the ATP from those two forms. Next step. Next thing. Nowhere on here, I know you're far away, I know it's small, I know you can't see it. You won't see CO2 anywhere up there, okay? There's no CO2 here. Here's why. When these hydrogens come flying back in, what the mitochondria does is it puts two of them together and adds them to that. Tell me what that big O is. Oxygen. So in the last step in electron transport, our waste product is actually water. That's what's made at the end. But do you think that our cells are going to waste the water that we make? No, we're going to use it. That's good, clean water that we're going to use. So this water right here, you may see sometimes me call it a waste product, and I might put it in quotes. So you'll remember, this is the equivalent of what we did with CO2. We used carbon to remove the oxygen. This time we're using hydrogen to attach the oxygen to keep that molecule to retain it, and then we can use it as water. I'm going to go ahead and go to the quiz. We'll answer the questions that we can, and probably I will give you answers to the others. Let's see. Number 19. How many CO2s were produced as a waste product during this step? 0. By the way, the fancy chemical reaction name for this step, we're using oxygen in the process, right? So we're going to keep that word oxidative. This time, we're not removing something, we're adding a phosphate. The fancy way, and you're seeing that right here on your handout in bold print, oxidative phosphorylation. That means we're using oxygen in this process. And this time what we're doing is we're creating or adding a phosphate. Sorry, we're adding a phosphate, and that is called phosphorylation. So sometimes this is called oxidative phosphorylation. Number 20. What is our waste product? Water. Very good. Okay, 21 and 22 you don't know, so I'm going to give to you. We have not mentioned this yet. We know we are making ATP, but we don't know how many. Okay, let me give you the key. For every NADH that goes through this process, we make about 3 ATPs worth of energy. About. It's not exact, but it's about. Okay, so you need to know the formula. NADH equals 3 ATPs. Okay, has enough energy to make 3 ATPs. Every single NADH, and we know we have a bunch of them. We have 6 from Krebs. We have 2 from glycolysis. We have 2 from that transition. Every single one of those can potentially be used to make 3 ATPs worth of energy. All right, the little guy, the FADH2, it's not as energetic. It's only used to make about 2 ATPs of energy. So that was the next answer. How many ATPs are produced from a typical FADH2? About 2. About 2. Was this process 24? Aerobic or anaerobic? Why would you call it aerobic? We used oxygen. Yeah, it's aerobic. Processing the whole thing. I know it's hard to do the first time you go through this. It's a bunch. What's the only step that we did not have to have oxygen? The first step, glycolysis. Every other step needed oxygen. The first step is the only anaerobic step. Okay, number 25. This is just a fun thing. I remember when I took national boards back in the day, they taught us this and they said it'll be on your national boards, and lo and behold, there it showed up for me. So I got a question right here. So I'm doing the same for you, whether you go into nursing or radiology or whatever. This is one of those crazy board questions that sciencey people like to throw out there. Okay, what is the final electron acceptor from this whole process? Well, watch me. Electrons were torn off and passed along the membrane. And when the hydrogen ions were pumped across, that H plus needed an electron to balance it, right? So the electron comes back to that as it's converted into water. And I don't know if you remember from a biology class or an AMP1, but water is a polar molecule. And so oxygen actually spends a little more time, the electron spends a little more time around the oxygen than it does the hydrogens. The fancy way to say it, instead of saying, oh, the electrons go to the water molecule, which they do, they say the final acceptor is the oxygen, because they spend just a little more time there. So the final electron acceptor in this electron transport chain is oxygen. It's oxygen. That would be O2. That would be O2. Okay. Ooh, what's the only thing on here that we talked about that could be called an enzyme and a pump? ATP synthase, yeah. The enzymatic pump that phosphorylates the ADP within the total deep within, they call this the matrix, within the matrix of the mitochondria, is ATP synthase. Okay, ATP synthase. All right. So what do we have left? Overview and, yes ma'am, go ahead.
Can you explain this electron transport chain process again?
Sure, absolutely.
Thank you.
So what organelle is this? The mitochondria. So, you know, I'm going to do this just to make it clean. I can put this back, okay? Oh, I think I can put it back. Will it change? Shouldn't change. There's the mitochondria, okay? There's our mitochondria. This, so look right here where I'm putting the pointer. That is the inner membrane that we did all the work on. Right here, right there, that's where the hydrogens get pumped to. So this is what we were showing in the other picture, just this little bit right here, okay? So let's go back. So there's our mitochondria. We produced, in all the other steps, we made some ATP. Everybody remember that? How many ATPs did we make in glycolysis, the very first step? 2. In the second step, we didn't make any. How many ATPs in Krebs? 2. So there's our ATPs. But we make all these other energy molecules. In the first step, we made 2 NADHs. Second step, oh, there's a pattern here. 2 NADHs. Third step, 3 times 2, 6. Third step, we made 6, okay? So now we have all of these NADHs that we have to process and turn into ATP. You might also remember that there's this little other one that makes energy called FADH2. We didn't make any, and then finally in Krebs we made a couple of them, okay? So we have all of these NADHs and FADH2s available to us. The only organelle that can process them and make them into energy is the mitochondria. So, fortunately for us, many of them were made in crabs, were made right here in the mitochondria. Okay? But the side, they just move in. So, all of these high energy molecules are in the mitochondria. How are we going to turn them into ATP? Let's make it as simple as we can. Two ways. We're going to take the hydrogens off, push them across a membrane, and create a gradient. We're going to pressurize it. Okay, let me go back to the balloon, right? Just like pumping hydrogen in, if I pump air into a balloon, I create high pressure in the balloon, right? Let me ask you, what happens if I hold the balloon here and I let go of the tail of the balloon? What happens to the balloon? You can see it right in your brain. What color's the balloon?
It's always red.
It was in my head, right? It was red. Okay. So there goes the balloon. See me pushing air into the balloon? That's energy. Your cell pumping hydrogen across a membrane? That's energy. So the first form of the energy is the energy on the hydrogens being moved across the membrane and eventually coming back. Okay? So first form of energy, hydrogens pumped across the membrane and eventually they move back. Second form of energy, the electrons get removed and get transported to various molecules. If you take biochem, you have to learn the name of every single one of those. It's so fun. And as it passes them along, it's transferring that energy that is kind of like electricity. And that membrane utilizes that energy also to energize that molecule over there, that ATP synthase. And eventually our electrons have to come back as our hydrogens come back. And we've made a water. But the true goal is not to make water. What's the true goal? To make ATP. What is the enzyme used to make ATP called? ATP synthase. And it gets its energy from two places, the hydrogens and the electrons. Did that help?
Yes.
With the big idea? So now we're going to do some details. I'm going to show you how to calculate all the ATPs from everything we did today. Okay? All the ATPs from everything we did today. So what we're going to do now is we're going to calculate the total ATP production. And we know most of that happens in the electron transport chain from those high energy molecules. But who can remind me? Glycolysis actually produced some ATP on its own. How many ATPs directly? Oh, as I put the number up there, great, thanks a lot. Two ATPs. So here's going to be our overall ATPs over here. And here's going to be all our intermediates. Glycolysis happened to make another molecule, a high energy molecule. How many of those did it produce? 2. Okay. Who remembers what our formula that we just learned by looking at the quiz was? Every single NADH converted into about how many ATPs? About 3. So here's how I do this. However many we have, I'm going to go times 3. And so that makes 6. So over here, this would lead to 6. Well, that's it. That's it for glycolysis. I'm going to abbreviate that. Oh, let's put decarb. At least we'll go that far. Okay. The next one, the reaction, oxidative decarboxylation. Did we make any ATP directly? No. All we made was what, energy-wise? And how many NADHs did we make? 2 in this step. Each of those 2 NADHs in a perfect world could be used to make how many ATPs? Well, not each one. 3. And so together, now tell me, what does that make? Okay. So there's 6 more ATPs. What was our third big step that takes place in the mitochondria called? Krebs. The big one. And in the Krebs cycle, get any ATP directly? Yes, how many? 2 ATPs. And, 2 ATPs stays 2 ATPs. It doesn't change. Oh, biochemists will tell me I just lied to you because sometimes that energy gets used somewhere. Krebs cycle is special because it not only produces NADH, it produces its little cousin, right? And let's just stick with our 2s right now to make this easy. I'm going to flip it from last time. How many FADH2s did the Krebs cycle make? 2. Very good. And those FADH2s, how many ATPs worth of energy does each one have in it? Only 2 ATPs worth of energy. And so what are our total ATP production from those FADH2s? It's only 4. But the Krebs is so special as the third step in the process. It makes a ton of NADH compared to everything else. How many did it make? 6. So don't you like all those numbers are 2 except for this? That makes it easy. Easier. Sorry, nothing easy about this. Easier. Each of those 6 can be converted into about how many ATPs? About 3 each. So 6 times 3. Wow. So in a perfect world, before biochemistry got exact and changed, this would be our overall ATP production if everything went according to maximal total production. I want to remind you, math doesn't have to be hard. When you're adding up all these numbers, play with me here. Watch this. Oh, let's stay away from red when we're doing math. What's 18 plus 2? 20, right? 18 plus 2 is 20. What's 6 plus 4? 10. What's 20 plus 10? There's 30. 38. Everybody see that? 38. So our total potential ATP production, if everything's cruising the best it could, but even back when I learned this the very first time in 1987 in biology class at Odessa College, they taught us that some cells in the body aren't as efficient and they only total 36. They just don't work as efficiently. You don't need to know what cells, anyway, so you will see when I ask a question, total production, sometimes I'll put 36/38. Just to give homage to that, to let you know that not everything always reaches maximum, okay? Biochemistry has got more detail, and now it says the NADHs and the FADH2s, they don't produce whole numbers of energy, so they've actually lowered your overall ATP production from a glucose down to about 32 is what they say now. But if I use that number, I couldn't show you the math and the concept wouldn't stick as hard. So I teach this and I just tell you guys, if you decide to go on and you go on and happen to take biochem or cell bio or some upper level science class that deals with the molecules, you will be able to understand converting this to their formula, okay? But this will serve you well in your programs for allied health because it gives you the concept of where those ATPs come from. And we think in whole numbers. We don't think in 1.8s and 1.2s and things like that, okay? So, the typical sugar, when it's broken down, can give you how many total ATPs? 36 or 38, right? Let me ask you this. What if you did your 100-meter sprint and you made your cell anaerobic? What step of the Krebs cycle would you be stopped at? Glycolysis. So none of the other steps could happen. Do you get that? And you wouldn't even have the NADHs because they gave up their hydrogens to pyruvate and became lactic acid. So if you didn't have oxygen available, how many total ATPs could you produce? Only the first two from glycolysis. Okay? So if you're anaerobic, you make 2. If you're aerobic, up to 38. So that's why my favorite question is number 30. Read number 30. I'm not going to read it. I'm letting you guys read this one. Well, it's variable. If you have oxygen, you only make 2. Oh, sorry. You can make up to 38. If you don't have oxygen, you can only make 2. So it's not always 2. It's not always 38. It is variable and depends upon the amount of oxygen that you have available in that cell. Okay. So D is the correct answer for 30. D. Oh, by the way, get this picture. Ooh, let me do one more thing. Let me just, I'm just going to put a couple of circles. Why? You'll tell me why. Oh, I don't need that arrow. Why did I circle those two? Ah, here's how I like to say it. You got it. Those are our direct ATPs. Those are our direct ATPs. There was no conversion needed for those two. Those two came directly out of the process. But all the other 34 had to come from those high energy molecules, right? We had to create them. That's crazy. Or 36. Or 30. Oh, I'm crazy now. I can't do math now. Okay. So our last couple of questions. Number 27. Cellular respiration includes each process except. Did it include glycolysis? Yes. Did it include a transitional phase that I called oxidative decarboxylation? Yes. Did it include Krebs? Yes. Was the electron transport chain important to this? Yeah, that's where most of our ATP came from. So the answer is all of these. Very good. All of these. 28. Most of the ATP molecules produced during cellular respiration are produced from which phase? Notice I didn't put electron transport this time. I used the other name. What's the other name for electron transport? Nice. Oxidative phosphorylation. Most of our ATP is indirect. Most of our ATP is in the electron transport chain, also called oxidative phosphorylation. Okay. By the way, most is a good word, but all of them but 4. All of them but 4 come from electron transport, also called oxidative phosphorylation. And then finally, number 29. In cellular respiration, how many ATP molecules are produced for each glucose molecule that gets completely metabolized when oxygen is present? 36 to 38. Very good. There is nothing easy about this process, okay? There are a lot of details. Most of your questions on your test, since we didn't have a lab and since this is very technical, will come right from here. You may have one or two questions that are based upon this, but are a little different, okay? But most of them will come directly from this quiz because it is so powerful to help you learn the process, okay? So this is our understanding of cellular respiration. Everything you need to answer test questions was done in our time together today.